Look, thank you very much for coming along. As you know, this is my last official lecture at Griffith. I'm sure I'm going to be giving research presentations later because my research team are staying here and I'm not moving some of my research. So, but I think it's important for academics maybe, to tell everyone what they're doing, what they're going, and give it, I think, an exit seminar. I went to a really good exit seminar, one of Jason's HDR cohorts, and I thought it was a good idea that I do the same. So what I'm going to do today is basically give you a bit of an overview of what's essentially 40 years of placental biochemistry. And I want to look back at some of the really important things we learned, some of the influences that have been influencing my research, and then give you an update of what we're doing now and what we're going to do in the future. So I haven't made it too, I'm in biochemistry, I haven't made it too biochemical because there's a variety of people listening online and here. So I'll try and keep it reasonably moderate. Okay, hope this works. No, it's not going to work. Great. Go over here. Now it'll work. So, some people in the audience will recognise this man. He was our biochemistry lecturer back in the early 80s, Frank Clark. He's an exceptionally good biochemist. He taught us just about, a few in the audience that know Frank, a really great guy. And I was having a beer with him in about 1982, so that's 40 years ago, and he started to tell me about placentas. I was a second-year student studying biochemistry, and I really didn't care too much until he started to tell me about embryo development, and he really got me into this. And I did a research project with him, and we did some good work together, and it ended up that Frank got an NHMRC grant based on the work that we did in my research project as a third year, and then he gave me a job for 3 years, which turned into a job for 6 years. And by that time, I'd fallen in love with placental biochemistry, and that was, I said, 40 years ago. So the placenta, I'll talk a bit more about the placenta in a moment, but the placenta really is the first organ that you form, and it's the most important organ you'll form because it dictates the development of every other cell in your body, okay? And so I'll talk you through that in a moment. But I just want to acknowledge Frank because, I think one day, hopefully, there might be a biochemist out there doing stuff, and they said, I listened to Tony back in the early 90s, and he was good, and it influenced me. You hear that, Lucy? You be quiet out there. Okay, so Frank was studying this thing called early pregnancy factor. Okay, so fertilisation in humans starts right up in the high fallopian tube, right up here. And it takes about 6 days for the developing embryo and then blastocyst to make its way down the fallopian tube into the uterus. Now, in that time, this little 2-cell, 4-cell, 8-cell, 16-cell little developing creature is emitting chemical signals which are developing the uterus ready for what we call the window of receptivity. Okay, so there's the adhesion molecules on the endometrial lining are changing, Velcro's going out ready to grab this little creature as it goes through. Immune cells are coming together because if that thing touches down, the immune regulation has to be right. It's semi-DAD, it's semi-allographic, and so it's part foreign, yeah? And there has to be a switch between TH1 and TH2. And I just found this fascinating. I said, this little beast, it's only 8 cells, 16 cells, 32, 64, 168 cells, and it's able to modify systemically mum's immune response and various things like the molecular repertoire on the endometrium. This is the first stage of differentiation from 64 to 128. And so these cells now on the outside of what's called the blastocyst are the trophectoderm, and they're going to turn into trophoblast cells, and that's your placenta. Okay, so that's the first cellular differentiation. This little blob in the middle here, which is just called the inner cell mass, that's you. That's all your other organs, your bones, your structures, your limbs, your ears, your eyes, your everything else. That's you. First differentiation is the formation of the trophectoderm and then the establishment of the placenta. We had this crazy assay going called a rosette inhibition test, and we could get changes in the amount of red cell binding to lymphocytes if we exposed them to bloods from mice. Mice mated about 4 o'clock in the morning. We could take a sample at 10 o'clock and tell whether that mouse was pregnant and had fertilisation. Yeah, so that's how early this thing was. So my first project as a reproductive biologist wasn't too flash. I used to go out to the abattoirs and collect sheep placentas by the drum. And I'd come back and I'd put them in a blender and I'd mash them up. And then I'd go chasing peptides that were able to give us some activity in this crazy rosette inhibition test. And that was my first job. And after about a year or two, I isolated a protein band on a silver stain gel. It runs at about a 25K dimer. You run it under reducing conditions, it comes down to 12K. This is 1986, 87, and we sequenced it. Back then, sequencing wasn't easy. We couldn't just send it off to someone to give you a protein sequence. We had to do a lot of protein chemistry to get it ready for sequencing. And we sequenced it, and we found out it was this molecule called thyroidoxin. That's my MPhil thesis. I did an MPhil whilst I was working. And so we characterised this. There was a couple of other players in here. I think one is platelet activating factor, and the other are a couple of weird eicosanoids. So we published a couple of papers back in 1987, and I worked for Frank characterising this system for about 6 years before going overseas. I was going through some data the other day, looking at old papers on EPF and I'm really interested in this again now because no one's really nailed this in terms of its characterisation. And I came across this paper written in 1981. Have a look at the last author. Alan bloody Cripps. You can't see, it's too small. Alan Cripps. For God's sake, Alan Cripps was writing about EPF back in the early 80s. And I worked for the guy for ages. I didn't even know that until recently. So there you go. So that was my first foray into placental biochemistry. I then went off to Oxford where I did my doctorate and did my postdoctoral studies there on placental biology. On a different area, I was working on stress axis hormones in the placenta. But I got this fascination then with the placenta because the placenta really defines how you're going to live your life. Around 84, David Barker started publishing work on the early developmental influences of fetal development and then associations with chronic diseases later in life. It's called the Barker hypothesis, but really underpins the developmental origins of health and disease. And things like submaternal or suboptimal maternal nutrition, stress in pregnancy, placental abnormalities, they change the way genes are expressed in the developing fetus, especially in that first 12 weeks. And then leads to altered growth metabolism, reduced birth weights, and then later in life predisposes the individual for things like type 2 diabetes, hypertension, cardiovascular disease. Yeah? So your in utero forms is really important to how you live your lifelong health. This is a whole area of science now. There are national, international societies on the developmental origins of health and disease, and it continues to sort of drive a lot of the work we do. Just before I go on, a little bit about placental anatomy, because I'm going to start showing you some pictures of cells. So this is a placenta. The placenta conveniently just plugs into your belly button. Some people think I've been doing 40 years of studying your belly button or belly button gazing because that's where the placenta plugs in. A little cord with a couple of umbilical veins and arteries, and then you have these cotyledons which basically protrude into the maternal endometrium. And then the gas and nutrient exchange happens across this cell layer of a syncytiotrophoblast, which is a fused multicellular layer, and the underlying cytotrophoblast. So the fetal maternal circulations don't actually cross over. Maternal circulation independent, fetal circulation independent, but the interface is between those cells. Okay? If you take that and do a cross-section, you'll see the syncytium on the outside, and this has been lit up with cytokeratins, and you can see the cytotrophoblast on the inside. Then inside here, I think I've got a better picture, inside here are fetal vessels. And then on the outside, the maternal blood pumps in and just sways it. Now, the best way to think about this is pick up a root of a weed and dust off the soil, and you'll see all the little roots. You know, the plant, you see all the little roots down there. That's just like a cotyledon in the placenta, and it's swimming in maternal blood. And so the maternal blood is supplying everything, and the fetus is harvesting that across that cell layer. So maternal blood here, and then fetal blood inside these capillaries. So I was interested in thyroidoxins, and I started to then look at thyroidoxin expression in these tissues. This is tissues taken from a first trimester pregnancy at week 12, where you have invading trophoblast tips coming out, and they're red hot for thyroidoxin. Yeah? But once this differentiation happens from cytotrophoblast through to syncytiotrophoblast, you lose expression. Okay? That was really remarkable to me as a placental biologist. Why is that switching off? Yeah, and we published this in an early edition of Placenta back in 1995. By term, in the villus, you don't see a lot of thyroidoxin expression. You see expression in the membrane. So this is an amnion, this is a chorion, and this is a decidual. We roll them up like a Swiss roll and chop them, and you'll see expression in the membranes, but it's switched off. They're the fetal vessels. You actually see fetal red cells in there that have been fixed, and then the maternal section's out, circulation's out here. So a couple of other remarkable things about placentas, okay? The first thing is that it's a total invasion. The embryo comes in, here's the inner cell mass now starting to differentiate. The blastocyst has been taken in, the epithelium of the endometrium is now resealed, and the trophoblast cells are starting to invade in here, looking for nutrition, looking for oxygen, but they're invading into the thing. Very much like a cancer invasion. Yeah, they switch on a whole bunch of tumorigenesis processes. They basically take over and drive through expressing metalloproteinases and various things. But they only go so far and then they stop. Yeah? There's a couple of different types of trophoblasts. There's these extravillous ones that come out from these structures. This is a villous tree. You can see the cytotrophoblast, and then there's an syncytium there, and then there are extravillous ones that invade out. And what they do, which I find really weird, is that they block maternal blood supply. They actually seal off the blood that's supposed to be supplying the embryo for about the first 11 weeks of development. So here's a spiral artery that's coming in, supplying blood, and this is caricature obviously, but you can do, these are real sections where you can see the plugs, the trophoblastic plugs have actually blocked the supply of oxygenated blood. Plasma is getting through, and the glandular secretions, the histotrophic nutrition that comes from the glands, the endometrium, is getting through, and that's what the embryos are surviving on. Yeah, you can map the pressure across this. So the pressure at 7 to 11 weeks, or 8 to 10 weeks, is down around about 20 millimoles of millimeters of mercury. And then after about 12 weeks, it unplugs. And Olivia and I, the first experiments we did together a while ago now, was to map the mitochondrial activity of placental cells across that 11, 12, 13, 14 week junction. And they change their metabolism. They go aerobic and they start to produce ATP. But in this early stage, then it's really quite anoxic, not anoxic, but hypoxic. And what that does, that is inducive to differentiation, cell differentiation. So all the cell differentiation, if you scan a baby at 12 weeks, you'll see toes, fingers, eyes, ears, everything is differentiated. That little cell mass at day 6, week 1, has turned into a little human being after about 10 weeks of gestation. Kind of crazy. What happens after that is then these plugs come out, oxygen comes in, the pressure, oxygen saturation comes back up, and you grow. Some more differentiation, but most of it has occurred. So this is really critical to how cells differentiate. If that doesn't happen properly, then generally there'll be a miscarriage. That's why the high concentration of miscarriages are in the first trimester, because that's the critical point of growth and differentiation. You think that's cool. I get really excited when mice can do the whole thing in about a week, yeah? And they have their whole litter in 3 weeks. So a little mouse can go from having sex to having a litter of 8 little perfectly formed mices in 3 weeks. That's pretty crazy cell growth and differentiation. Okay, now the other thing we learn about all this is that this doesn't happen properly, and you don't get this remodeling of these spiral arteries. So the trophoblasts have got in here, they've blocked it off for about 11 or 12 weeks, then they've deplugged and they're letting blood come back through. If that doesn't happen, then you get really bad perfusion. You get constricted vessels pumping blood at high velocity into this space, whereas if it happens properly, you get low velocity, high volume blood being pumped in. And that's one of the key features of a disease called preeclampsia. Okay, and so the last 30 years or so, we've been spending a lot of time looking at preeclampsia and trying to understand its pathophysiology. So I'm going to tell you a little bit about preeclampsia. Okay, it's reasonably prevalent. It's not 7%, it's about 4%. It's still killing a lot of people per year, about 80,000. Generally occurs after 20 weeks gestation. You start to see changes in blood pressure, in hypertension, so systolic blood pressure and diastolic blood pressure. You start to see edema, you start to see protein leak into the urine. They're the classic symptoms. If you don't intervene, then you get this thing called HELP syndrome, which is hemolysis, elevated liver enzymes, low platelet counts, and then you'll get eclampsia, where women will go into a full clonic-tonic seizure and basically die. Yeah, so we intervene now. We say clinicians intervene. They'll generally deliver the placenta. Now, if you take the placenta out of the uterus, then generally this condition resolves. It's a placental disease. The origins of the placenta origins of preeclampsia start in the placenta, although a few of us like to debate this about late onset and early onset, and we can talk about that later. But so we've been interested in this pathophysiology of this disease. So we know that it starts with poor endometrial inner placental junction myometrial preparation and then poor invasion of the myometrial spiral arteries. No changes in vasculature underpins this disease. You get poor placental ischemia reperfusion injury happening, or poor perfusion coming in, and then you get markers of placental oxidative endoplasmic stress. What that does, then I'll take you through some of the biology of that, it starts to release components into the circulation, things like soluble flit and other mediators. The maternal endothelium becomes activated. It's like a systemic immune response. You get hypertension, you get leaky endothelium, and then you start to get the clinical symptoms of the disease. But this is one of the key features that underpin it. Our focus is on placental oxidative and endoplasmic reticulum stress. So I've been explaining all that for a reason, because it gets me back to these enzymes called thyrodoxins and thyrodoxin reductase. So I came back to this university I came back to Australia in 95 and I started working down here in 96. In 95, I can only think of about one or two people in the world who were talking about this concept of oxidative stress. Barry Halliwell was one of them and Hans Seiss was the other. And I got kind of excited about this because I was working with redox active proteins in the placenta. And so working with thyrodoxins, I started working on thyrodoxin reductases, glutathione systems. Over here, these are the nasties that are generated during aerobic respiration. We generate superoxide, it's converted to hydrogen peroxide. You can make hydroxyl radicals and various other radicals. And you can see markers of oxidative damage in placentas. You can see oxidized lipids, oxidized DNA, oxidized proteins, And there's this thing called oxidative stress. Now, the world's got right into this. I think we published back in 1995. Now there's over 1,000 papers on PubMed, if you put in the terms, placental oxidative stress and preeclampsia. So one of the first things I did in my lab, once I got a lab established at this campus, it took me a while to get a lab established at this campus, was to look at the protein expression. These are activity assays, the activity of these proteins in normal and preeclamptic placenta. So we looked at thyrodoxin, we looked at thyrodoxin reductase, we looked at the lipid peroxides, which are the oxidized form of lipids. We looked at glutathione peroxidase, we looked at superoxide dismutase, and we looked at protein carbonyls, which are the oxidized form of proteins. We had 18 normal placentas, and then we matched, we had 20 abnormal placentas, and we matched those with 18 normal placentas. And you can see that we're seeing physical evidence of oxidative stress. The antioxidants are down in each case. The oxidized biomolecules are increased. So we published this in Placenta in about 2003, I think, from memory. We then went chasing the protein expression levels. Well, I've shown you this slide. There's no thyrodoxin in that outer syncytium. A couple of groups followed that up with other antibodies, different antibody techniques, and you can see there's no thyrodoxin. This is a really nice picture from this Japanese group. There's no thyrodoxin being expressed in the syncytium. The underlying cytotrophoblasts still have a full repertoire of antioxidants. They're mononuclear cells. My mate, Les Myatt, who works in Portland, published this in 97, and the quote is that neither manganese superoxide dismutase nor copper zinc superoxide dismutase appear to be present in syncytiotropoblast. Les couldn't afford the colour micrograph, unfortunately, and published it in black and white. But there's no SOD. A Japanese group again looked for the mRNA expression of thyrodoxin reductase. Another quote, syncytiotrophoblasts showed almost no immunoactivity for any thyrodoxin reductase antibodies. So there's none of the reductase there. And then finally, Heighton Mystery, working in Nottingham, published a series of really good studies on glutathione peroxidase. And again, there is very little glutathione peroxidase expressed in the syncytium of these term sections. And he, like us, found that this enzyme is essentially wiped out in this disease called preeclampsia. So it really raised the question then, is this antioxidant expression in that fused tissue essential for its biology? Does it make it vulnerable to oxidative stress? So you've got the syncytium here. The cytotrophoblasts are fusing to form the syncytium. Is that normal? Is that a normal part of cell fusion and cell turnover in the placenta? Is it? Well, we know it is worse. Why is it worse in preeclampsia? And can we do something about it? Can we prevent it? Can we intervene? Okay, to do something to come in here and maybe make that a bit better. So, a little bit more about preeclampsia. This is a micrograph or an illustration from a paper published in 1893. That's 1893, okay? I didn't get it wrong, 1893, by a guy called Small, who was a pathologist working in Germany. And these are illustrations of pieces of placenta in the lungs of mothers who died from eclampsia during childbirth. Okay, they're pieces of tissue, multinucleated bits of placenta in the lung tissue. And he was asking why these women who have a neurological thing, they've got multi-organ failure, but they've got a neurological problem because they're going into chronic seizure and dying, why was finding bits of placenta in the lungs? What we know is that this is a series of turnover events, okay? So the cytotrophoblasts fuse in the syncytium, and they're there for a period of time, probably 2 to 3 days, and then the nuclei, you can see multinucleus, changes in mitochondrial morphology. I'll show you some work that Josh Fisher did. And then you have release of this material into maternal circulation. So maternal circulation is bathing the outside of the placenta, and the placenta is epithelial. It's like your skin, it's constantly shedding. 2 to 3 grams a day of material is coming off the surface of the placenta into maternal circulation. Now, the macromolecular stuff gets cleaned up in the macrophages in the lungs. Some of the molecular then, free DNA, other proteins go to the maternal liver, and a couple of cycles around the blood supply, it's all wiped out and it's all normal. Yeah? And it's generally through an apoptotic process. You can get multinucleated, I'll show you some blood slides in a moment. You get multinucleated bits of trophoblasts, you get mononucleated bits of trophoblasts, and you get little fragments, you get all sorts of garbage. We just call it garbage, placental garbage coming into maternal circulation. But in preeclampsia, then the mitochondria are changing. You don't get the full energy production that you need to basically go through the apoptotic process. You get necrosis, you get aponecrosis, you get all this stuff coming into the maternal circulation. And it's immunogenic. Yeah? It's stirring up mum's immune response. So that picture on the top is a part of my PhD thesis from the John Radcliffe. I can see a syncytial knot here. So this is a bit of, on the syncytium, on the surface of a preeclamptic placenta. You can see this is about to bud off. Marion, not Marion Knight, the other Marion Johansson, working at the John Radcliffe, was doing blood smears, taking smears of uterine bloods from preeclamptic women. And you can stain for trophoblast and you'll see multinucleated, mononucleated. Marion Knight was working on other fragments of what we call STBM, basement membrane fragments from the trophoblast. You see high concentrations of those in preeclampsia. And that's the surface under electron micrograph of the surface of the placenta in preeclampsia. You see all this garbage coming out. And so you get fetal DNA, fetal proteins, exosomes, lipid rafts, all potential antigens, all 50% DAD, all likely to cause an immune response, all elevated in preeclampsia and causing endothelial activation so that you get the symptoms of the disease. So back to this question, can we do something about it? At the time, this is the number of papers published on oxidative stress and antioxidants from 1995, when we started to get into this, to 2008. The field has just been gone through the roof. Yeah, it's just everyone thinks that oxidative stress is the cause of everything. I could do another lecture about oxidative stress and why I don't think it's the cause of everything. But it's just gone through the roof. And antioxidant therapy is part of this. Yeah, so people got into these antioxidants using vitamin C, vitamin E. It's worth a lot of money. You look at the number of published papers on oxidative stress. You know, the number of clinical trials have been done, and the same happened in our field. So between about 95 and 2005, there were about 50,000 women treated with vitamin E, vitamin C in clinical trials, RCTs, designed to prevent preeclampsia. None of them worked. Yeah, one, they chose the wrong vitamin E, and there has not been, this is a quote from Barry Hallowell's book on free radicals and biology and medicine, which is kind of the Bible for anyone that works in this field. Sadly, intervention trials with antioxidants have yielded a morass of confusing data. And that data for reproductive biology, so there was a study in Canada. We had one, Gus Decker and Caroline Crowther did one down in South Australia. There's one in Lucilla Poston and crew did one in the UK. There's one in Brazil. James Roberts did one in the US, and there was another one in the UK. Essentially vitamin C, vitamin E, administered mid-gestation, 20 weeks through to term, and didn't have any clinical effect really in stopping it. So very disappointing. But we had another angle, because a lot of the proteins we were playing with are selenoproteins. Okay, they have selenium in their active site. So selenium sits here. I should have brought the periodic table, Pete and Hannah. They've given me this beautiful periodic table as a going away present today, and I should have brought it over here and put it on the table. In the middle there, you'll see selenium. And selenium is critical because it forms selenocysteine, and selenocysteine is in the active site of 25 proteins in the human body. Okay, only 25. It's not a random process. It's a misread of the UGA codon. And this is the 21st amino acid, protogenic amino acid, which is selenocysteine. So these are the 25 selenoproteins we've been playing with. Okay, we haven't done them all, but we've done a lot of work on GPXs in the placenta. We've done a lot of work on thyroidoxin reductases. We've done a little bit now on deodonases. We look at thyroid function in pregnancy, and the deodonases convert T3 to T4. They're important in... We've started to look at some of the endoplasmic proteins with those lovely people who are online there somewhere, Nat and Tue, and Lucy's working with them down in Melbourne. We had a girl down there last year, Claire, doing a master's project looking at some selenoprotein in placenta, some of these unusual ones. So we're trying to explore whether or not this is a important mediator of placental function. So let me just take you through these are cell studies that we have been doing over the years. So the good thing about playing with selenoproteins is that if you increase the amount of selenium in the cell culture, and you can grow trophoblast cells, if you increase the amount of selenium from 50 nanomolar, 100 nanomolar, 200 nanomolar, 400 and 800, you can increase both the expression and the activity of enzymes like glutathione peroxidase and thyroidoxin reductase. Okay, so it's a lovely system. You don't have to knock things in or knock things out. You can just change the micronutrient. And there's no selenium in in culture media anyway. It's just in the bit in about 12 nanomolar in the fetal calf serum. So you can just engineer this system to express the proteins in this system, yeah? So it's a really neat way of doing it. So that's what we do, is that we grow cells, and this is work that Alicia Kira did in our lab when she was doing her PhD with us. So we grow different types of placental cells, swans, jegs, BWOs, with and without sodium selenite or selenomethionine. You can use the organic form, selenomethionine, or you can use just sodium selenite, it's cheap. You can use that as well. Increase the amount of GPX of thyroidoxin reductase. You then come along and block complex 1 or complex 3 with things like antimyosin or rotenone. Rotenone blocks complex 1, and you'll generate reactive oxygen species, and you can measure them. So this is a DCF assay, increasing amounts of rotenone, and you can measure the increase in superoxide as it comes out, produced by these cells in culture, because we block their complex 1 and the electrons are being leaked to oxygen. If you pre-treat with sodium selenite or selenomethionine, you can block that DCF response. You can also increase cell viability, so we run things like MTT assays that Elsa's been doing, and you can increase the viability of these cells by protecting them with selenium, and you can block apoptosis. We look at things like BCL2 and the apoptotic mechanism. So we can provide cellular protection with a simple trace element in vitro in a lab, I must say, but it seems to work. Yeah, that's good. At the same time, we started to do mitochondrial function work. So we're doing oxygraphs, and we're putting these cells into the oxygraph. Now, what an oxygraph does, it has 2 little oxygen probes in here. So you're growing cells in these 2 little chambers, and these are oxygen probes, and it measures very precisely the amount of oxygen used per time. We can measure picomoles of oxygen per second, okay? And it's just the mitochondria using it. You can put tissue in there, you can put isolated mitochondria in there, you can put cells in there. These experiments are done with cells. If you put selenium in, we used BWOs, JEGs and SWANs. If you put selenium there, then you increase the respiratory capacity of these cells to breathe, their mitochondria to breathe. Yeah? And that's the trace. So you can see different things we do to this. We engineer the electron transport chains. So we can block complex 1, complex 2, complex 3, etc. And then we can engineer it. And you see this green trace is with selenium and this red trace is without. And so if you turn that into picomoles of oxygen used per second per cell, then you can see that selenium is good for cells to breathe. Now, at the time we were doing this, we spotted this paper. It's done with a different cell type. These were hippocampal neuronal cells from mice, done by an American group. But they were seeing the same traces, almost identical. Yeah, and they published that if you add selenium, then you get really good respiration in these different type of cells in a different model. And that got us really thinking about, well, I wonder how this is working. And they mentioned a protein called selenoprotein H. I'd never heard of it, but we went chasing it. And so we went chasing it in the placenta and in cells. And if you increase the amount of selenium and you probe for selenoprotein H, you can see that in BWOs, JEGs, and SWANs, selenoprotein H expression goes up in both sodium methionine and sodium selenite. And then if you look for PGC1 alpha or NRF1, you'll also see overexpression. Now, what are PGC1 alpha and NRF1? They are regulators of mitochondrial biogenesis. Okay, they're the key regulators of mitochondrial renewal and mitochondrial biogenesis. So now we had some evidence to say that not only is selenium protective against oxidative injury, it may actually be beneficial in regenerating mitochondria. And you can show that, okay? So we can look at mitochondrial number by mapping the gene expressions of nuclear genes to mitochondrial genes. There are 13 proteins, there's 30-something mitochondrial genes. So if you do mitochondrial gene ratios to the nuclear gene, you can estimate the number of mitochondria in a cell. And that goes up when you add 100 nanomolar of sodium selenite or selenium methionine at 500 nanomolar, which is the red bar. And we came along then with a green, it's not actually green, it's clear, but it's a drug called aranophen, which binds selenocysteine and knocks out selenoproteins, and we can reverse that effect. Okay, so we're pretty clear that I think this is a mitochondrial biogenesis effect. And you can actually map it. You do we haven't done confocal, but this under light. You put mitotracker in, and you'll see increased mitochondrial expression in these cells after exposure to selenium. So our conclusion from all this work is that selenium is good for mitochondria, yeah, placental mitochondria. It increases the expression of these important antioxidant selenoproteins, reduces ROS production, improves viability during oxidative stress, decreases mitophagy, improves mitochondrial respiration, increases the amount of selenoprotein H that's expressed, and that in turn drives mitochondrial biogenesis. So this was all good fun, lots of papers, lots of interesting work, but can we take it to any further to maybe a clinical interface? So we moved into animals, and we've done lots of work with animals on selenium-free diets. We have exercising animals. If you put animals on selenium-free diet, a normal little mousie, when he gets going, he can run about 7 or 8K a night. If you take the selenium out of their diet, they only run about 5 or 6. Yeah, they lose their ability to exercise. We had spontaneously hypertensive rats that kept dropping dead from heart failure because they had no selenium in their diet. That was an interesting experiment. We set it up, ran it for 2 years, and at the end, we got really interesting. They all started to die. And then we had pregnant ones. And the pregnant ones, if you take pregnant rats and then you put them on diets for 4 weeks, get them pregnant, then you'll see if you measure the enzyme activities of GPX and thyroidoxin reductase in the liver, you can wipe out the activity of these proteins. What happens to their pregnancy? Well, they get high blood pressure, they get hypertensive, they get proteinuria, no change in pup weight or placental weight, significant weight, but you get increased markers of oxidative stress. So by taking this micronutrient out of the diet, everything else about the diet's equal. By taking it out of the diet, you can then cause a in vivo, if you like, animal model of preeclampsia. So at the time, Jess Vanderlei was doing this work. At the time, Jess and I were thinking about how the globe's selenium levels are different the selenium level in country is dependent upon the soils. The selenium comes through the soil into the food chain. That's how you get your selenium. So for a bit of fun, we went and looked at all the literature published on selenium and rates of preeclampsia. We found 189 papers. And so we plotted them. This is the plasma level on micrograms per litre of selenium, Egypt, Nicaragua, New Zealand pre-93, Finland pre-93. They're all important numbers. Then there's this incidence of preeclampsia in these populations. There are 11 million pregnancies in this database. Yeah? And this is the correlation. You can see that if you've got high levels of plasma selenium, you've got low levels of preeclampsia. And if the levels of preeclampsia, the levels of selenium in the diet and the population is very low, then the reported incidence of preeclampsia is very high. So this was starting to get more evidence that selenium might be important in pregnancy. 95 micrograms per litre is the cut. If you have less than 95 micrograms per litre in your blood, you are seleno deficient. If you have more, you're seleno replete. Yeah? If you put that cut across those 45 countries and you put the 24 countries in with the below 94, this is the preeclampsia rate. It's about 4.4, 4.5, 4.3, 4.4. If you look at the countries that are over 95, it's down to just below 3. Yeah? Circumstantial evidence, but still the plot's thickening. 2 countries in the world recognised in the 90s that they had a problem in their public health because of low selenium. One was Finland and the other was in New Zealand. In 1994, Finland legislated that all fertilisers used in the farming had to have a certain amount of selenium in it. And they did that, and in New Zealand they put it in the food chain because they do a lot of feedlotting and feeding animals, and they put it in the food chain at the level of animal supplementation. So this is what happened to the selenium levels in Finland pre-94 and post-94. It went up as a population, and this is New Zealand. The preeclampsia rate went down from 3.8% to 1.9% over that time period in Finland, and went down from 6.5 down to below 2 in New Zealand. So again, pretty circumstantial evidence, but interesting. So then we got us into doing a whole bunch of work with selenium and pregnancy. And there's a lot of data out there. These are a bunch of studies. They've shown that you need to have more selenium in your diet. If you look at the NH&MRC RDI for selenium, it increases by 10 micrograms per day for women who are pregnant and lactating. So even our national authorities are recognising this trace element is absolutely essential to a healthy start to life, and it's recommended that women take more. Decreased plasma selenium in recurrent miscarriage has been published. We're seeing journals now. This study by Maria back a few years ago had 33 different types of studies. And then there's also this other evidence, well, I'll come to in a moment about this. So around this time, I was still collaborating with my buddies over in Oxford, and they started a selenium intervention trial. There'd been a couple done, one in China, one in Iran, but Chris Redman and Margaret Raymond put together an intervention trial called the SPRINT trial, Selenium and Pregnancy Intervention Trial, ran from 2010 to 2012 at the John Radcliffe. There was 200 high-risk women, half got a placebo, half got selenium, 60 micrograms per day, quite a low dose, 100 micrograms per day is generally the supplement you would use. And it was applied quite late in pregnancy. It was post 20 weeks. I think you're going to do this properly. And then we got involved by measuring some of the plasma and GPX and soluble FLIT and endoglin, these markers of preeclampsia. Now, the outcome of this is this was really poorly underpowered. Okay, the incidence of this disease is 4%. You know, if you wanted to power a clinical intervention to try and see changes in this, you'd probably need 10,000 women plus. But it did show changes in the levels of soluble FLIT and placental growth factor, which are the key markers of preeclampsia. So we started to see biochemical changes and biochemical interventions or biochemical modifications by this trace element intervention. And that got us into this wonderful world of trace elements and metabolomics. So we started measuring selenium in whatever we could get our hands on. We'd measure it in wine, we'd measure it in orange juice, we'd go to the supermarket, we'd buy antioxidants. We haven't measured it in coffee yet, but we will measure it in coffee if you want to. And Dan McKeating, who's not here, he said he was going to come today, but he must be busy doing something else. I bet he'll be at the pub, for sure. he got involved, and then he started to measure 2 trace elements, 3 trace elements, 4. We got up to 27 trace elements in 100 microlitres of blood. So we can take one sample with Will Bennett, who Richard knows pretty well, the guys over at Smart Water, 100 microlitres of plasma, and we can get 27 trace elements, which are all those trace elements there. So not quite the whole periodic table, but half of it. And then we started mapping those trace element levels against diet and against pathologies. And it's been really interesting. Nothing really has jumped out and grabbed us and said, look, that trace element's absolutely essential. Why? Because I think everyone's diet's different, everyone's genetics different. So when you start doing metabolomics, it gets really quite difficult. And you can imagine if someone has a Gatorade drink just before they go to clinic, these aren't fasted samples, okay? Or some of the samples are fasted, but not most of them aren't fasted. So there's this background variability based on people's diets, breakfast, genetics, so it's difficult. We also got into small molecule metabolomics. So we measure hundreds of small molecule metabolomics. This one here is, there are some 34 in this data that's related to lipid metabolism and amino acid metabolism and carb metabolism. Again, Lucy, who's working down in Melbourne, and Dan, who will be at the pub, are still working on this. And Hannah's done some work on this too. This is sort of where we got to in 2021 with some of this data. First thing I said to Dan, right, okay, if you've got all that metabolomics data, show me the profile of a non-pregnant woman versus a pregnant woman, and can metabolomics cluster those two together? So these red, this is the women up here, these are all these small molecule metabolites. So up here you've got things like valine, isoleucine, methionine, cysteine are there, various types of small molecule metabolites. These are the non-pregnants, and these are the pregnant. So only one of those women didn't cluster into that group. So we felt pretty good about this, that we can actually use metabolomics to tell if someone is pregnant or non-pregnant. Then we started to look at these are preeclamptic, term, and fetal growth restricted. Okay, again, you can see the preeclamptics are clustering pretty well. The fetal growth restricted are a bit more spread, and the terms are over here. So not too bad. We took 6 of these elements and we built an algorithm that Hannah and Dan have done, and we can pick up with 97% sensitivity and specificity preeclampsia in this cohort of samples. My buddies in Melbourne, who are behind me somewhere, went and validated this in another cohort of samples, and it all went to SHIT. It didn't work in another cohort. So still the jury's out, So we still think metabolomics might have a role to play, but it's confusing, and it's a really, you're working on a background here of variability in diet and genetics, which is really difficult to deal with. And of course, overriding that are all these women who are using supplements. Okay, we do have some information about that, but the supplement use in Australia is also confusing this. So I'm going okay for time. I talk quickly, I know, I get excited about this. I'll put it out of the podcast, you can listen to it in your leisure and go through it again. Okay, what are we doing at the moment? And what's the work I'm currently working on and the work I'm going to be working on, even though I'm going to be doing a Dean's gig again. So we're still working on these key placental research questions. What is causing placental dysfunction? What's going on in placentas that aren't working optimally, and why aren't those babies growing as they should with that really important organ? Can we measure it? Can we detect it? If a baby's born prematurely and goes into an intensive care bed wired up to a whole bunch of machines, you can just go and read the machine. The machine will tell you exactly what's going on. It'll tell you its oxygen level, heart rate, blood pressure, etc. All there. Great machines, excellent. Placenta, no dials, can't see. How do we read the signals coming in the placenta saying that the oxygen levels aren't good in that baby, or what's going on? And finally, can we do something about it? Can we intervene to, we're talking really, a lot of these diseases, you start to see them around gestation week 28. Preeclampsia, you don't really see it before week 24, 23. We're talking about a 10 to 12 week window of buying some time to optimise fetal delivery. A baby born at 38 weeks has a completely different developmental trajectory, and, babies at 38 weeks are done. 37's term. They're fit and healthy, ready to live in the world. Babies at 28 weeks are vulnerable. Babies at 25 weeks, it used to be babies at 25 weeks, well, that was the minimum, that was the cutoff. But now, you're seeing babies around the world being born at 23, 24 weeks because neonatology is so good. So what are we currently working on? I've got these beautiful people to work with now and into the future. So I've been at this campus 26 years and it's only been the last 18 months that I've finally got a group of people together who are able to get daily access to pregnant mums and get a really good clinical scientific interface going. So Fabrizio De Silva Costa here, I've known Fabrizio a long time, but at the beginning of last year, he moved from Brazil to the Gold Coast. He's the head of feto-maternal medicine at the Gold Coast University Hospital. He's brought in Carmen, who's a feto-maternal medicine specialist from Hong Kong. Okay, I've just got to pause for a moment. Come on in. Good timing. Welcome, Jessie. So I was just about to talk about you. And then Fabrizio and the university and the Hospital Foundation have supported 2 research midwives. So now we've got clinical teams on board who are able to actually get access to mums and bring us samples on a daily basis. So this has been working great. Then over across the road are these lovely team of scientists. Olivia's been with me a while. Jessie, welcome. Aaron, and then Hannah and Chloe, who are doing their PhDs. And so we've got a really good team set up now where we're getting access to clinical samples and we've got all the clinical data and we started what we call the placental health study. So we're recruiting mums at 13 weeks gestation. Normally mums wouldn't go to a clinic until they're about 18 weeks pregnant. We're recruiting mums at 13 weeks. They're coming to clinic and we're doing scans. We're taking a lot of maternal histories and then we're taking samples. We take plasma, sera, PAX tubes for RNA, DNA, and urine samples, and we're biobanking them back in GO5. Yeah? Now, why is that important? So if you take maternal histories and then put in mean arterial blood pressure, you add in uterine pulsatility index, you add in PLGF, placental growth factor, you add in soluble FLIT, and you put these into an algorithm, you can predict 100% the incidence of early onset preeclampsia. So we can detect early onset preeclampsia. Now that's preeclampsia that occurs before 34 weeks. Unfortunately, only about 20% of preeclampsia happens that early. Most preeclampsia is over here towards term and late onset, yeah? And our predictive ability then is not so good. Okay, we're down to about 45%. So our mission impossible, if you like, I should play the music, is to find metabolites, biomarkers, and microRNA or RNA species in either exosomes or maybe free in the maternal plasma that we can add into this equation to bring this up to have diagnoses of preeclampsia later in pregnancy. These are early samples, yeah, okay, so this is prognostic. We're taking these at 13 weeks. These women come back at week 28 and we see them again at week 36, then we see them at term. That's our new toy. It's about $300,000 worth of hormone analysis equipment called a Cryptor. Fabrizio pulled off a deal with Thermo Fisher. We've got it for the next 3 years for free. It arrived week before last, and we've got all the reagents to analyse. We're on our path to recruit 2,000 mums in this study. We started in May we started, and we've hit 100, so we're going pretty well, yeah? We've also got a couple other birth cohorts on the go. One is the My Body, My Baby that Janelle did, where she recruited mums when they come in for their glucose tolerance test, and we've got a lot of information. We've got trace element data, we've got dietary data, we've got metabolite data for those mums at a single time point. And then we've got the Queensland Family Cohort, which is run out of the MARTA. We've got about 500 mums, bubs and dads, lots and lots and lots and lots and lots of data on stress, economics, all sorts of stuff. But then we've got biological samples, blood, breast milk, cheek swabs, everything from 24, 28, 36 weeks. And we started to do analysis of that. We've done trace elements, we've done metabolomics, and we're starting to do some analysis of that data as well. Come back to selenium. One of my sabbaticals, I went to Oxford in 2005, and I was sitting there talking to Chris Redman, who's kind of a bit of the guru of preeclampsia. And I was getting really excited telling him about selenoproteins and GPX and thyroid oxygen reductase. And he turned around and he said, have I told you about my toenails? And when a senior prof at Oxford talks about his toenails, you kind of get a bit worried, you know? And what Chris had been doing, he'd been collecting toenails from pregnant women who were suffering from preeclampsia and from normal pregnancies. Now, when you analyse toenail, you cut that nice little bit off the end there and you analyse it for selenium. You're actually analysing selenium level 3 months prior, because it takes 3 months for that piece of tissue to grow from there to there, and that's where it's laid down. So he had data on selenium 3 months before disease onset, yeah, in 2005, and I thought I was being clever. And that's been backed up now. There's been a publication come out, it's in 2019, on risk of early pregnancy, and these are week 13 samples. So I don't think selenium's out of the picture yet, but we'll be able to tell when we get some more placental health study samples. Okay, so most of you in the room know me as the head of school or the dean, and by the end of 2018, I'd had enough of being a dean. Some are in the room going, why do you want to go do it again? So I took 2019 off and I went to Cambridge for a year. Okay, now the big buzz then was organoids. Okay, this is the Science front cover, organoids, the 2018 breakthrough of the year. And there was a young lady working at Cambridge who had worked out a way of taking placental cells at week 10 and getting them to grow in 3 dimensions in organoids, because we grow cells in 2 dimensions, and that's not really how they work. So I went off to Cambridge for a year, or most of a year, to study mitochondrial biology. Now, when when cells fuse, you need to go. Yeah. Okay, I won't point you out. As cytotrophoblasts fuse across into the syncytium, their mitochondria change totally. They go from looking like grapes to looking like raisins. They shrink. They drop their ATP production by about 40, 50%. They change their stereogenesis potential. They change their biology. This is what Josh Fisher was doing when he was in my lab. And so we were looking at models of studying mitochondrial transformation. What happens to these mitochondria? Are they still functional? We know they're producing progesterone and pregnenolone, changing their ATP synthesis. They're not dead. And so we're looking for models of this. And so we set these organoids up, and I was able to grow some of these organoids down here. These are mine. You can stain them for mitotracker, and they differentiate in the middle to form a syncytium, and they change their mitochondrial respiration. And we've got some nice videos. Josh came over and worked with me for a while in Cambridge. Got some nice videos looking at mitochondrial changes in the middle of these organoids. Now, a young lady called Georgia Kafer works at the University of Sunshine Coast, got an NHMRC grant last year to study placental organoids, and lucky me, I get the job up there, then I can go and talk to her. She came down and talked to us in January because she's having a bit of a struggle getting a lab built up there for her work. She's now working at the University of Queensland until they build a lab up at the Moreton campus. George is a little bit excited because she gets another placental biologist to come and play at the Uni Sunny Coast with her. But I'm going to be working on this. The diagnostic work that we're going to do with the hospital stay here with Olivia and team, and I'm going to be working with Georgia on organoids and mitochondrial function again. Okay, so hopefully at some point in this lecture you've come to a realisation that the placenta is the first organ that you have. The first cell differentiation is trophectoderm. Here's a placenta. For those of you who haven't seen them, they're about bread and butter plate size, about this thick, 600 or 700 grams. It's not the prettiest thing. You know, most people throw them away. They're really interesting culture around placentas, what people want to do with them. This is on the front of the Time magazine years ago about how the first 9 months of life changes how you have your lifelong health. But the thing that placenta does is really remarkable. You know, it's your lungs. It provides your oxygen, provides your nutrients, takes away your waste. Okay, it's turning over all the time. It's supporting it. Your life support system, conveniently plugged in here. All the circulation that goes on in the body when the blood's coming in under passive sort of processes. It's just fascinating how that works. It produces hormones, it protects immunologically, it does all sorts of stuff. So it's pretty amazing. So just to finish off, I want to pay some acknowledgement to some remarkable people who have been through my lab over the last little while. So this picture, I promise all the time never to use this picture again, but this picture was taken just after I was appointed as Dean at Health here in about 2013 or 14, somewhere around there, because Olivia's in the picture and she was, and all these other people were PhD students. And they weren't very happy because I was going off to be a dean and I wouldn't have time to supervise their projects. Now, I just want to tell you about a few of them. So, you know, Dan McKeating, he's just come back from Cambridge. Pierre's just about to finish medicine at Wollongong. He'll be a doctor in a couple of weeks. He's just on his last clinical rotation. Josh is a postdoc down at the University of Newcastle. Anna went off to medical school. George is still around. Janelle is now a research fellow. She's got a Royal Australian O&G fellowship and she's working up in Cairns as a midwife, and she's still collaborating on our data. Jess Vanderlelly, who was working here then as a lecturer, she's now the Deputy Vice Chancellor of Students at La Trobe University. Olivia, not there. Yeah, where? Oh, you're there. Yeah, sorry. Oh, you're there. And then Lucy is working as a postdoc down in Melbourne with our colleagues down there. And so this is my current team. Guys, we will continue to do work, and I'm sorry I won't be here every day, but I'll be here every couple of weeks. And I just want to acknowledge the team over across the road. So thank you for coming along. It's been a hoot. I'm not going to stop doing research. It's what I started doing before I was doing anything else, really, and I'll continue to do that. But in the meantime, as I had a phone call this morning from one of my seniors at the University of Sunshine Coast, and they said, enjoy your last day of freedom. See you Tuesday. Thanks, everyone. So the plan is now, you can ask a question if you like, but the plan is now we're just going to probably bingle across and go to the bar and talk about placentas. So. So Lucy's put something in the chat. Congratulations. Did anyone want to ask a question about placentas? Jen? Yeah. So they have a higher chance of developing vascular diseases later in life. Yeah, there's something about their endothelium which is not quite right. So they have higher incidence of cardiovascular diseases and chronic diseases related to hypertension later. Mums as well. So if a mum has a preeclamptic pregnancy, her chances of getting cardiovascular disease and hypertensive type syndrome later in life go through the roof. Yeah? So something changes in the endothelium kind of permanently due to that stimulus during pregnancy. Yeah, we don't understand exactly how it works, but you know, a lot of the data now is really, so our medical record keeping the last 30 or 40 years has really improved. Yeah, and so now women who might have had a preeclamptic baby in the 1950s or 60s, now you're starting to see those women come through into their 80s, 90s, or 70s, 80s. You're starting to see chronic diseases. Yeah. Darren?
Is there any genetic background, like the baby?
Yeah. Yeah. There's a genetic component. There's kind of three levels of genetic components. Yeah, there's individuals. So you'll see mothers who have preeclamptic children. The children have a higher incidence of preeclampsia. These are sort of, no one's nailed it down to what gene. No one's done a genome association study, or there's been some markers around angiotensin, renin type markers, but no one's nailed it really. The second is the compatibility between male and husband and wife, or I shouldn't say that, partners. Because you'll see women who have a normal pregnancy and then they change partner and have a preeclamptic pregnancy. So there's a compatibility, and vice versa. You'll see women who have preeclamptic pregnancies or even infertility change partners and it goes back to normal. Yeah? So there's a couple of different levels that are working in the background there. But no one knows exactly what. Not that I'm aware, no. What, sorry? Oh yeah, it's different. I think placentophagy. Yeah, eating a placenta is a really interesting phenomenon. Yeah, in different cultures around the world, back in the dark ages, used to have different attitudes. Most of you know that I breed cattle. I had 3 calves born last week. How's that? I got beautiful little calves. But each of those mothers ate their placenta. Cows will turn around, the first thing they'll do, they'll lick the calf, then they'll eat the placenta. And they are herbivores, they shouldn't be eating that meat. And they'll eat a skerrick of it. And why do they do it? Well, some people think that consuming placental placentophagy is good for mental health and prevents the postnatal blues. Yeah. Other people think it's really important in inducing prolactin and bringing on milk. I think in the animal kingdom that's the case, because the first thing that cows need to do is to feed their calves colostrum. Colostrum is the first milk that calves get. It's full of antibodies, full of growth factors. If calves don't get it, they struggle for months. Yeah, they've got to get it, and it's a really viscous material, and they've got to get that through their long teats and have them probably fed for 8 months. So I think it's got something to do with getting that first milk supply into the calves. Yeah, sheep do it, horses do it, dogs do it, but dogs are dogs eat anything anyway. I don't know what cats do, but yeah, cows really weird. They'll turn around and eat their placenta. I think biologically there definitely is, because but sociologically now there are people over at the university hospital who come in and they'll take your placenta, they'll freeze dry it, they'll powder it, put it in capsules. You can take it home and take it for a price. You all right? Yeah, go, Elsa. The original, I guess, there's a background. Trophoblasts have to invade, yeah, and the invasion. No. So the placental cells have to invade in, yeah. Now, if the compatibility between partners, and especially the expression of the HLA axis, HLA-G has to be expressed for an appropriate immune response. Now, if you don't get the right compatibility between the invading trophoblast and the mum's immune system, then you've got problems, yeah. But the real thing is the invasion of those spiral arteries. If you don't get those spiral arteries eroded by the trophoblast, then you don't get the correct blood supply, and then you get perfusion changes in the placenta, and that causes then release of things out of the placenta, which causes mum's endothelium to go crazy, and that's really this disease, yeah. But it comes down to the compatibility of and the ability of those trophoblast cells to invade in. Oh, is there? Sorry, was anyone else? Yeah, Steve? Good question. There is a theory in my head that, and other people I've talked about, is that women who have a mitochondrial predisposition to poor mitochondrial function, this is the first major physiological challenge, yeah? The first time they get a major is growing another baby inside them, and then they come up against this problem and their mitochondria aren't dealing with it, and that leads to placental insufficiency and fetal growth restriction. Yeah, and later on, the next biggest problem is maybe when they're later in life and their cardiovascular function is not so good. Josh Fisher, who is at Newcastle, there are 30-something mitochondrial genes. 13 proteins are synthesised in the mitochondria from mitochondrial genes. The other 1,200 proteins that are in the mitochondria come from nuclear genes. Josh has got a theory that the compatibility between genomic expression and transcription, translation, and then mitochondrial trafficking into the mitochondria for those proteins, if that's not right, then you're going to have a mitochondrial predisposition. Yeah? And he's got an NHMRC ideas grant that I'm CIC on in consideration at the moment, looking at GWAS studies, looking at compatibility of maternal mitochondrial markers with placental mitochondrial markers. So we don't know, but it's a really good question, and it could be. Yeah. And I just reviewed a grant for a group in Oxford looking at mitochondrial supplementation to take mitochondria from white cells of mother and put it into placentas, not in humans, in animals. Yeah. So people are looking at mitochondrial supplementation and right way to boost mitochondria. We did it from a selenium point of view because that's what we thought would work, but we still don't know it works in a big population. Okay, look, 2:05, I'm past pub time. We can talk about placentas. Thank you very much. I sent an invite to 200 metabolism students yesterday, to come to my 4 to 6 lecture online that I did last night. I got 7. So we put out an invite. Thanks, Fran, for organising today. We put out an invite to you guys. Look, I know there's half a lecture theatre. It's so good. Excellent. Thank you. Thanks, Lucy.
Congrats, Tony. Enjoy the bar. I'll see you in New Zealand. Take care, everyone.
Okay.
Thank you.